Hello students, I am Dr. Vinod Shinde. In the previous lectures, we have discussed about EMP pathway and HMP pathway for the metabolism of microorganism. For energy generation, microorganism metabolize glucose in different pathways, that is EMP, HMP. Now we will discuss about the next pathway which was utilized by microorganism to metabolize glucose, that is ED pathway or Ettner-Dodorf pathway. This pathway was discovered by Ettner and Dodorf in 1952. This was a pathway of metabolic studies of Pseudomonas saccharophila, which has been observed in many other species of this genus, that is Pseudomonas, and it was observed in Pseudomonas saccharophila majorly. And this is the first is three reactions of ED pathway are identical to HMP shunt or HMP pathway, hexose monophosphate pathway. And it was observed that due to, however, not certain whether or not enzyme differ in the kinetic characteristics, the pathway was run by organism of Pseudomonas species, Pseudomonas genus, sorry. The other species of Pseudomonas genus also carry this pathway as a uh, pathway uh, to be run in the critical conditions. Glucose in this pathway, glucose was utilized by the microorganism in different manners, giving you uh, energy consumption, low energy production, and more energy consumption. But this is the survival pathway of the microorganism. Glucose in the first step, glucose is utilized or reacted with enzyme hexokinase. In the presence of hexokinase, giving you glucose 6 phosphate or phosphorylation reaction occurs as in the EMP pathway. In the regular senses, the glucose to glucose 6 phosphate and glucose 6 phosphate goes to the EMP pathway in the regular senses. But if you go further, in the absence of enzymes for EMP pathway, we have glucose converted into glucose 6 phosphate, ATP utilized and ADP produced, and glucose 6 phosphate is utilized to deglucono delta lactone 6 phosphate, giving you similarity toward second reaction similarity towards the HMP cycle. The same enzyme glucose 6 phosphate dehydrogenase was used, producing giving you utilization of uh, enzyme glucose 6 phosphate dehydrogenase, utilizing 6 glucose 6 phosphate to deglucono delta lactone 6 phosphate, producing NADPH2. 6 hydrogen electron transport chain occurs here. So in this deglucono delta lactone 6 phosphate goes to 6 phosphogluconate or 6 phosphogluconic acid in the degluconolactonase enzyme, presence in the presence of degluconolactonase enzyme, similar to the third reaction of HMP cycle. Here, if HMP the enzymes for HMP pathway are available, then we will go to the organism will go to the HMP pathway. But if HMP pathway is not present, or EMP pathway and HMP pathway are absent, that is enzymes for EMP and HMP are not available, then the organism tend to divert towards the ED pathway that is observed in the Pseudomonas saccharophila or Pseudomonas genus specifically. Phosphogluconate dehydrogenase enzyme is uh, acted on by uh, is act, 6 phosphogluconate was acted on by phosphogluconate dehydrogenase enzyme, giving you 2 keto 3 deoxy 6 phosphogluconate, that is KDPG, 2 keto 3 deoxy 6 phosphogluconate. This 2 keto 3 deoxy 6 phosphogluconate was acted on by KDPG aldolase, that is 2 keto 3 deoxy 6 phosphogluconate aldolase, This enzyme splits the 2-kdPg into pyruvate and glyceraldehyde 3-phosphate. The pyruvate is the ultimate product, so no further reaction occurs with pyruvate. The glyceraldehyde 3-phosphate reacts with fructose 6-phosphate and gives you, in the presence of trans ketolase enzyme, erythrose 4-phosphate and xylulose 5-phosphate. Xylulose 5-phosphate converts into ribulose 5-phosphate in the presence of ribulose phosphate epimerase enzyme. The ribulose 5-phosphate was produced. Erythrose 4-phosphate again reacts with fructose 6-phosphate, giving a 10 carbon and aldolase enzyme, which splits the 10 carbon into pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate again mm, react with each other or combine with each other in the presence of transketolase enzyme. Ketolase, aldolase, transaldolase combination reaction occur here also, giving you xylulose 5-phosphate and ribulose 5-phosphate. If you observe carefully here, the last two substrate, the last two components, that is xylulose 5-phosphate and ribulose 5-phosphate, are observed in HMP pathway in the forward reaction. Here, if you reverse the ED pathway, you will observe that this is the reverse of the HMP pathway in some sense till the glyceraldehyde 3-phosphate and fructose 6-phosphate. This was observed in the HMP cycle. Xylulose 5-phosphate, ribulose 5-phosphate, pseudoheptulose 7-phosphate, glyceraldehyde 3-phosphate, fructose 6-phosphate, erythrose 4-phosphate, xylulose 5-phosphate. This all is observed in the HMP pathway. This is the some kind of reverse of the HMP cycle. So this was observed in the Pseudomonas saccharophila, and it was run by Pseudomonas genus only. In this, we do not get that much energy, but this is the survival pathway of the organism. So, in the next lecture, we will discuss about the pentose phosphate pathway, or phosphoketolase pathway, which has pentose and hexose phosphoketolase pathways. Two types of phosphoketolase pathways are there, which are observed. So in the next lecture we will discuss that, and this is the ED pathway, eternal order of pathway. So thank you.